In molecular biology, ribosomal ribonucleic acid is the RNA component of the ribosome, and is essential for protein synthesis in all living organisms. It constitutes the predominant material within the ribosome, which is approximately 60% rRNA and 40% protein by weight. Ribosomes contain two major rRNAs and 50 or more proteins. The ribosomal RNAs form two subunits, the large subunit and small subunit. The LSU or RNA acts as a ribozyme, catalyzing peptide bond formation. rRNA sequences are widely used for working out evolutionary relationships among organisms, since they are of ancient origin and are found in all known forms of life. Structure: the ribosomal RNA is complex with proteins to form two subunits, the large subunit and small subunit. mRNA is sandwiched between the small and large subunits, and the ribosome catalyzes the formation of a peptide bond between the two amino acids that are contained in the rRNA. A ribosome also has three binding sites called A, P, and E. The A site in the ribosome binds to an aminosyl tRNA. The amino group of the aminosyl tRNA, which contains the new amino acid, attacks the ester linkage of peptidyl tRNA, which contains the last amino acid of the growing chain, forming a new peptide bond. This reaction is catalyzed by peptidyl transferase. The tRNA that was holding on the last amino acid is moved to the E site, and what used to be the aminosyl tRNA is the peptidyl tRNA. A single mRNA can be translated simultaneously by multiple ribosomes. Prokaryotes versus eukaryotes. Both prokaryotic and eukaryotic ribosomes can be broken down into two subunits, Nt equals length in nucleotides of the respective rRNAs, for exemplary species Escherichia coli and human. Note that the S units of the subunits cannot simply be added because they represent measures of sedimentation rate rather than of mass. The sedimentation rate of each subunit is affected by its shape, as well as by its mass. The Nt units can be added as these represent the integer number of units in the linear rRNA polymers. Prokaryotes in prokaryotes a small 30S ribosomal subunit contains the 16S ribosomal RNA. The large 50S ribosomal subunit contains two rRNA species. Bacterial 16S ribosomal RNA, 23S ribosomal RNA, and 5S rRNA genes are typically organized as a co-transcribed operon. There may be one or more copies of the operon dispersed in the genome. rRNA contains either a single rDNA operon or multiple copies of the operon. The 3' end of the 16S ribosomal RNA binds to a sequence on the 5' end of mRNA called the Shine-Dalgano sequence. Eukaryotes. In contrast, eukaryotes generally have many copies of the rRNA genes organized in tandem repeats. In humans approximately 300–400 repeats are present in 5 clusters. Because of their special structure and transcription behavior, rRNA gene clusters are commonly called ribosomal DNA. The 18S rRNA in most eukaryotes is in the small ribosomal subunit, and the large subunit contains 3 rRNA species. Mammalian cells have 2 mitochondrial rRNA molecules and 4 types of cytoplasmic rRNA. The 28S, 5.8S, and 18S rRNAs are encoded by a single transcription unit separated by 2 internally transcribed spacers. The 45S rDNA is organized into 5 clusters on chromosomes 13, 14, 15, 21, and 22. These are transcribed by RNA polymerase I5S occurs in tandem arrays, the largest one on the chromosome 1q41-42. 5S rRNA is transcribed by RNA polymerase III. The tertiary structure of the small subunit ribosomal RNA has been resolved by X-ray crystallography. The secondary structure of SSU or RNA contains 4 distinct domains: the 5' central, 3' major and 3' minor domains. A model of the secondary structure for the 5' domain is shown. Translation. Translation is the net effect of proteins being synthesized by ribosomes, from a copy of the DNA template in the nucleus. one of the components of the ribosome base pairs complementary to a shiny urodalgano sequence upstream of the start codon in mRNA. Importance of rRNA. Ribosomal RNA characteristics are important in medicine and in evolution. rRNA is the target of several clinically relevant antibiotics: chloramphenicol, erythromycin, casugamycin, micrococcin, paromomycin, ricin, sarsin, spectinomycin, streptomycin, and thiostrepton. rRNA is one of only a few gene products present in all cells. For this reason, genes that encode the rRNA are sequenced to identify an organism's taxonomic group, calculate related groups, and estimate rates of species divergence. As a result, many thousands of rRNA sequences are known and stored in specialized databases such as RDPII and Silver. Recently, rRNA have been shown to be the origin of species-specific microRNAs, like miR663 in humans and miR712 in mouse. These mRNAs originate from the internal transcribed spaces of the rRNA. Genes: RPL1, RPL2, RPL3, RPL4, RPL5, RPL6, RPL7, RPL8, RPL9, RPL10, RPL11, RPL12, RPL13, RPL14, RPL15, RPL16, RPL17, RPL18, RPL19, RPL20, RPL21, RPL22, RPL23, RPL24, RPL25, RPL26, RPL27, RPL28, RPL29, RPL30, RPL31, RPL32, RPL33, RPL34, RPL35, RPL36, RPL37, RPL38, RPL39, RPL40. RPL41, MRPL1, MRPL2, MRPL3, MRPL4, MRPL5, MRPL6, MRPL7, MRPL8, MRPL9, MRPL10, MRPL11, MRPL12, MRPL13, MRPL14, MRPL15, MRPL16, MRPL17, MRPL18, MRPL19, MRPL20, MRPL21, MRPL22, MRPL23, MRPL24, MRPL25, MRPL26, MRPL27, MRPL28, MRPL29, MRPL30, MRPL31, MRPL32, MRPL33, MRPL34, MRPL35, MRPL36, MRPL37, MRPL38, MRPL39, MRPL40, MRPL41, MRPL42, RPS1, RPS2, RPS3, RPS4, RPS5, RPS6, RPS7, RPS8, RPS9, RPS10, RPS11, RPS12, RPS13, RPS14, RPS15, RPS16, RPS17, RPS18, RPS19, RPS20, RPS21, RPS22, RPS23, RPS24, RPS25, RPS26, RPS27, RPS28, RPS29, MRPS1, MRPS2, MRPS3, MRPS4, MRPS5, MRPS6, MRPS7, MRPS8, MRPS9, MRPS10, MRPS11, MRPS12, MRPS13, MRPS14, MRPS15, MRPS16, MRPS17, MRPS18, MRPS19, MRPS20, MRPS21, MRPS22, MRPS23, MRPS24, MRPS25, MRPS26, MRPS27, MRPS 28, MRPS 29, MRPS 30, MRPS 31, MRPS 32, MRPS 33, MRPS 34, MRPS 35. These denote genes encoding for the proteins of the ribosome and are transcribed as mRNA, not rRNA. See also, ribotyping. References. External links, Silver RNA Database Project and LSU, European Database on Small Subunit Ribosomal RNA, Ribosomal Database Project 2, 16S rRNA, BioMineWiki, page for small subunit ribosomal RNA, 5 feet domain at RFAM, ribosomal RNA at the U.S. National Library of Medicine. Medical subject headings.